Okay, so in this next section, we're going to look at different organelles inside the cell. Organelles are going to be structures inside animal cells that typically have some function. Now, one thing to understand is that we won't always see all of these organelles in every type of cell. For example, let's take the endoplasmic reticulum as a good example. We will see a lot of endoplasmic reticulum in cells that require packaging and organizing proteins. However, there are cells that don't necessarily require that. Adipose cells are a good example, so we're not going to see a lot of that. There are cells that have a ton of mitochondria, muscle cells. Not surprisingly, they need that. There are other cells that don't require mitochondria at all. They don't require a lot of energy, which is what mitochondria play a role in. And so understanding, though we're going to go through all these organelles, not every cell has all of these. Some have more than these. Some cells have more than one nuclei. So just kind of understand that as we go forward. And this is sort of a basic overview of each of these we're going to look at. Obviously, you can memorize this table. I'm not going to go through this table because we're going to handle each of these individually. But this is sort of just a table of contents, I guess, sort of a path of where we're going to go, looking at these various organelles. I'm going to try to do this as hopefully as simply as we can. Again, as I said before, there are other videos on Canvas for you that'll go through some animations of these, how they work. So take a look at those. Let's start with the easiest one being the nucleus. Now, the nucleus is, we often think of as kind of the brains of a cell, and I guess that's not a bad analogy. It is the largest organelle generally in cells. And the nucleus literally is almost its own entity. It has its own nuclear membrane, it has its own DNA, it contains chromosomes, it has its own nucleolus. There's also chromatin in here as well. And so we could argue that the nucleus is, it is where a lot of the function of the cell, what it does, is going to originate from. If the cell's job, for example, is to make a certain hormone, like pancreatic cells, like beta cells in the pancreas, producing insulin or glucagon. Not surprisingly, the nucleus is going to play a key role in making sure DNA is formed to form those proteins. That may not be the case necessarily in other cells, like neurons, that don't produce proteins per se, but they do have other functions. And so the nucleus is going to be a key organelle. One thing to understand is that there are going to be some cells that have multi-nuclei. Muscle cells are a great example. Muscle cells have typically more than one nucleus. Skeletal muscle cells. They're called multi-nucleated. And so understand that when we get to muscle cells, we'll see that a little more clearly. And so understanding that the nucleus is going to play a key role. A second organelle, and I guess this is sort of an organelle in a way, is the cytoplasm. So think of the cytoplasm as the everything else in the cell. We do technically consider it an organelle. It's basically going to be in the material that's going to be between the nucleus and the plasma membrane. Now, obviously, there'll be other organelles in here, mitochondria, endoplasmic reticulum, ribosomes, lysosomes, things like that. But there also will be fluid. Now, what's key, we will see some types of cells that have actually a lot of structure inside the cytoplasm. A good example of this, actually muscle cells are a good example of this. There are actually unique structure inside cytoplasm to give muscle cells a little more form, a little more structure. We see this in bone cells, osteocytes as well, where we'll actually see inside we'll actually see some filaments in here that give this cell in the cytoplasm, more overall structure, more rigidness to it. Other cells, for example, like immune cells, we generally won't see that. So cytoplasm, we will also see other things in here, like other molecules, compounds, and gases would find themselves in the cytoplasm as well. All right, so let's move on to the mitochondria. Mitochondria, for our conversation, may be one of the most important parts of the cell because it is here that we see the production of what's called ATP. ATP is adenosine triphosphate, and it is the key form of energy for cells to do what they need to do. Great example would be muscle cells. Muscle cells have a ton of mitochondria. Well, why? Muscle cells have to contract. That contraction requires energy. So not surprisingly, we have a lot of mitochondria. So mitochondria are very unique. They actually have their own DNA. There's actually some thought in some scientists that mitochondria actually originally were bacteria or something like that, and then animal cells just use them and for their own purpose, and so they're actually maybe have different types of origin. We're not going to get into that, but they do have their own DNA. There are going to be cells that replicate more mitochondria if need be. Mitochondria are also unique in that they have this double membrane. They have an outer membrane and an inner membrane. If you take exercise physiology with me or some more advanced physiology classes, we go into the importance of this. That actually this inner membrane, kind of like the inner club of the mitochondria, is where a lot of ATP really get produced using oxygen. And so that inner area and this membrane that separates the inner and the outer mitochondria is really, really key. We call these cristae, these folds here. There's some ribosomes here as well. And so this mitochondria is going to play a really key role. We're going to talk a lot about mitochondria. You'll see them in lab on the muscle cell, and I believe on the neuron, the nerve cells as well. And when we talk about muscle cell contraction, we are going to talk quite a bit about mitochondria. And so just understanding these are going to play a key role in how ATP is formed, and ATP being formed from basically what we eat. So that might be carbohydrate, which is a good choice, but could also be fats or proteins as well. And so that's converting these carbon and hydrogen bonds to something that's more readily available, to utilize to form ATP. Okay, so let's move on. Mitochondria, we're going to go now to the endoplasmic reticulum. So the endoplasmic reticulum is unique often to different cells. And so maybe the best way to describe the endoplasmic reticulum is to use an analogy of a factory, where in a factory you have an assembly line where goods are manufactured and then packaged and ultimately sent out and shipped to wherever they need to go. If you kind of think of that analogy, the endoplasmic reticulum would be the manufacturing. This is where maybe proteins, maybe glycoproteins, lipids, whatever they are, would be synthesized. This typically being directed by the nucleus and what's going on in nucleolus. And so think of this as sort of a conveyor belt in a way of synthesizing, manufacturing whatever the cell needs to manufacture. And then it'll travel down this Golgi apparatus and begin to transport through the endoplasmic reticulum, and then it'll work to transport these out to the Golgi apparatus, which we'll talk about in a second. So ER is going to play a big role. We'll see later on that there are two different types of ER. There's smooth endoplasmic reticulum. There's rough endoplasmic reticulum. Basically, the difference is rough has ribosomes. This should say ribosomes, not no ribosomes. There we go. So that's going to have, rough will have ribosomes, smooth will not have ribosomes. They do have some other functions. You'll see rough more in some cells than others. Don't worry so much about that. But that we will see these. And in fact, in lab, in the animal cell, you'll actually see both of these. There's actually smooth ER and rough ER, which isn't always common, but for your animal cell, it'll work. So just understand there's a difference between these two and what they do, what they function. The next is a Golgi apparatus. And again, if you kind of work through the same analogy of what these cells do, this would be more of kind of the packaging and beginning the process of transporting whatever has been manufactured. And so typically the Golgi apparatus has a somewhat similar look as far as endoplasmic reticulum without the ribosomes, potentially. Similar kind of overlapping shape. But these are going to play a role typically to transport and begin to go with the process of getting rid of excreting, secreting, whatever this is, protein, lipid, what have you, outside of the cell. And it's going to use secretory vesicles to exocytose. That's a term we use to basically get rid of out of a cell. There's membrane vesicles as well that'll do this. You'll see this on your animal cell models. And this is where lysosomes can potentially play a role. Lysosomes, typically they're going to carry enzymes. They can break down things. They can also transport things. And so this Golgi apparatus, really cool name, named after the scientists who discovered those, can play another key role here. And last, and certainly not least, are lysosomes. Now, lysosomes, we're not going to get into a ton in this class until we get to the immune system. But lysosomes are going to play a key role on two fronts. One is they are going to play a key role in the immune system in other cells to basically phagocytose, or lack of a better word, eat various viruses, bacteria, stuff that needs to be dealt with. And so we see this, the example here is a white blood cell, but that's not the only cell that could potentially do that. Lysosomes can also package, instead of breaking down, these things can also package and break down carbohydrates and like glucose and proteins as well if the cell requires those. So lysosomes typically have enzymes here that'll assist in breaking down whatever it needs to break it down. It plays a key role in some types of cells that we will talk about later on in the semester. So that's it as far as some of the organelles. The next thing we're going to talk about is how do we transport things in and out of the cells, how that's done, and different mechanisms to which we're able to pull that off.